Hello everyone, I'm Guillaume Nesa, the co-founder and the CEO of Holit. Holit is a French company created in 2013, based in Aix-en-Provence in the south of France with 10 employees. Expert in human breast analysis, Holit has a know-how in the design of miniaturized non-dispersive infrared gas sensor. We started our first development by trying to solve a societal problem in terms of road safety and public health with alcohol consumption. Alcohol is the leading cause of road deaths in Europe, about 50,000 deaths a year and several hundred thousand injured. Alcohol-related deaths are preventable and must be reduced with the right tools. Assessing, measuring, and monitoring are the keys to good alcohol management. After 6 years of R&D and industrialization, we designed our first product, Osigo, a reusable and connected breathalyzer that integrates our miniaturized infrared spectroscopy technology. In this presentation, I will present the main technological challenges that we faced during the development of our infrared spectroscopy sensors. NDIR technology is based on the principle of infrared light absorbed by a sample at a specific wavelength. Our sensor consists of a cuvette crossed by an infrared light to a detector. The molecular composition of the target gas causes an absorption of energy at a specific wavelength. This attenuation is measured by the detector combined with a bandpass optical filter that eliminates all other wavelengths that the selected gas molecule may absorb. Finally, the gas concentration is calculated according to the Beer-Lambert law. NDIR systems are commonly used for their reliability because the wear and the drift are very negligible compared to other technologies. Another benefit of this technology is accuracy because there are few interferences with other gases than the one targeted. But NDIR spectrometers are generally bulky, non-mobile systems, and not dedicated to analyze samples with high humidity as the hexanal air. Miniaturization and portability of these spectrometers were a real challenge. Indeed, according to the Beer-Lambert law, the longer the optical path, the more sensitive the system is. Developing a miniaturized and mobile spectrometer to analyze compounds in the hexanal air required to manage the accuracy of the measurement and the electrical consumption of the system. By combining high-performance components, algorithms, and specific designs, we came up with a miniaturized NDIR sensor dedicated to breast samples analysis. First, we developed a patented cuvette that offers a heating system and a high optical reflective layer. By combining these two features, we obtain a cuvette that allows the analysis of high humidity samples while maintaining the accuracy of the measurement as described in the Beer-Lambert law. In addition, the detector and powerful algorithm allowed us to obtain a sensor with a detection threshold of around 20 ppm for hexanal measurement. Another benefit of the cuvette is its low thermal inertia. It allows a startup time of about 30 seconds and a saving of electrical energy. The areoolic sampling was designed by simulation to comply with the physiological differences between individuals and avoid thermal disturbances. Finally, the infrared source has been designed and managed to optimize the infrared radiation intensity and the electrical consumption. Osigo is the result of 6 years of research and testing. It is one of the best performing breathalyzers on the market in terms of accuracy, reliability, and robustness. Osigo integrates OC Engine NDIR sensor and a rechargeable battery into a compact design. It complies with the European and North American standards for electronic breathalyzers. It can even indicate false positive results by detecting residual alcohol in the mouth. As you can see on the graph on the bottom left, we can monitor optical signal at a sufficient frequency to determine the integrity of a breath sample. Osigo also offers a long-term reliability, avoiding any calibration for 18 months. Breath analysis is one of the non-invasive methods that may be used by most people and allows high-frequency tests. To meet this aim, the upcoming technology has to be easy to use, reliable, mobile, and bring fast results. Volit has developed innovative miniaturized NDIR sensor with high sensitivity, specificity, and reliability. Our perspective is to go further to detect and measure other volatile organic compounds in hexanal hair thanks to our NDIR technology. Either breast metabolites after drug or substrate administration, or endogenously produced breast compounds due to a particular physiological state, could be measured with such systems. Our first explorative results focused on the analysis of carbon dioxide, one of the most representative markers of a person's metabolic state in exhaled air. Indeed, this gas can be a marker of respiratory disorders, but can also allow other applications such as knowing the fertile period of a woman. We have already succeeded in adapting our OC engine sensor for CO2 analysis, and we are now focused on new gas analysis. Thank you for your attention.
Thank you for visiting this poster entitled Changes in Exhaled Volatile Organic Compounds Following Iron Supplementation in Self-Reported Healthy Adults. I am Nabeetha Nagalingam, Principal Translational Scientist at Allstone Medical Limited. It is my pleasure to review the findings of this study, which was done in close collaboration with the Functional Gut Clinic. Here we investigated the effects of iron supplementation on healthy adults. Iron deficiency anemia is a global problem affecting more than 1.2 billion people. Right here in the UK, it accounts for about 13% of gastroenterologists' referrals. Iron deficiency anemia can be treated with iron supplementation, either intravenously or orally. However, unabsorbed iron can cause gastrointestinal symptoms such as bloating. This is believed to be associated with intestinal microbiome changes induced by the iron treatment. These intestinal gases, including hydrogen and methane, can be absorbed by the intestinal tract and into the circulatory system. They can then be transported to the lungs, where they can diffuse through the gut-lung lining and be exhaled. Breath tests can then detect these gases. Tests for detecting changes in the intestinal environment can be facilitated by using fermentable carbohydrates such as lactulose. Lactulose can be metabolized by intestinal bacteria rapidly, producing gases within 180 minutes. The time at which these gases are detected can be linked to regions in the gut where the bacterial metabolism is most active, thus providing insight on how iron treatment affects the regions along the gut. In this trial, 25 healthy volunteers provided breath samples collected in breath bags following lactulose ingestion. Figure 1 shows that samples were collected at 4 time points: at 0 minutes, 45 minutes, 90 minutes, and finally at 180 minutes post-lactulose ingestion. Over the following 28 days, volunteers ingested 200 mg ferrous sulfate tablets daily. At day 28, volunteers repeated the lactulose test, providing breath samples at the 4 time points mentioned above. Breath bags samples were all sent to Allstone Medical Labs in Cambridge and analyzed using SIFT-MS technology. Targeted data were interrogated for statistical significance. In Figure 2, it is shown that the exhaled compositions in ambient samples were different from those in breath, and that the 2 ambient samples, one taken at the collection site in Manchester, which is called the room ambient, and the other at Allstone Labs in Cambridge, called the lab ambient, are both different from each other. There is also some overlap with the breath samples and the room ambient, which will be discussed in the conclusions. Table 1 shows that among the compounds investigated, 7 showed significant changes after 28 days of iron supplementation. This was recorded at time 0, before the lactulose challenge. In Figure 3, 3 compounds were further investigated: propanoic acid, acetic acid, and hydrogen sulfide. The first 2 compounds significantly increased after 28 days of iron supplementation following lactulose ingestion. This is in comparison to the day 1 samples, which were taken before iron ingestion. Highest differences were observed at 180 minutes. This indicates that iron supplementation was associated with microbiome changes that were most prominent in the colon. Also, both propanoic and acetic acids are short-chain fatty acids which are considered beneficial in gut health. Contrary to this, hydrogen sulfide significantly decreased over time following the lactulose challenge. This indicates that iron supplementation induced changes in the small intestines and not the colon. Hydrogen sulfide is usually considered to be detrimental to gut health. You can hear more about hydrogen sulfide in the BBCon talk, The Past, Present and Future of Breath Testing for Bacterial Overgrowth. A limitation of this study is the high background signals in the room ambient, as mentioned earlier. This may be due to a number of sources, including cleaning agents and perfumes. Future experiments should be carried out in a room where the air is filtered. Nonetheless, all the changes that were found to be statistically significant were at least 2 standard deviations from the lab ambient. Taken together, despite these limitations, iron supplementation in this cohort appears to be beneficial. Further investigation on how this data relates to signs and symptoms of the volunteers will be done to understand the effect of iron on this population. Thank you for your attention, and we are happy to answer any questions at breathbiopsy@altstone.co.uk.
Hello everyone, myself Manohar Prasad Bhandari, researcher at the Institute of Clinical and Preventive Medicine of University of Latvia in Riga. The title of my poster presentation is Volatilomic Patterns of Gastric Juice and Their Potential for Diagnosis of Gastric Cancer. This study was carried out as part of the Horizon 2020 BOGAS project and a postdoc Latvia project. A bit of background. The study of volatile organic compounds is an easy and economic approach for the non-invasive diagnosis of disease. This study is focused on the volatilomic signatures or the VOCs of gastric juice obtained from gastric cancer patients and healthy controls. The main objective was to characterize the chemical patterns formed by the VOCs released by gastric juice by using gas chromatography, mass spectrometry, and to evaluate if the cancer-related alterations could be employed for gastric cancer detection. Experimental section. The study cohort included 35 patients and 58 controls from different populations, including Ukraine, Brazil, and Colombia. The samples were collected during upper endoscopy by sucking gastric content. A minimum of 3 ml of samples was taken into 20 ml glass vials. Similarly, blank sample containing 10 to 12 ml of distilled water was also taken to identify the contaminants. The collected samples were stored and transported at -80 degrees centigrade. For the GCMS analysis, heady-space solid phase microextraction as a pre-concentration method coupled with GCMS using azylent system was used to identify the VOCs released by gastric juice samples from gastric cancer patients and the healthy controls. The extraction temperature was maintained at 70 degrees centigrade, incubation time 60 minutes, and the extraction time 3 minutes. Now on to results and discussion. The profile of GCMS chromatogram identified different volatile organic compounds from gastric juice samples. A total of 1,181 distinct compounds were identified in the gastric juice head space. Out of these compounds, 6 of them were occurred in at least 80% of the samples. They were hexanal, benzaldehyde, ethyl acetate, acetone, ethanol, and pyridine. Some of the compounds, such as 2-propanol, propofol, hexafluoroisopropanol, and their metabolites, they arose from the hospital environment or the sample collection site, ambient air. Some of these compounds could also arise from the GCMS instrument itself. Now, studying the distribution of the compounds according to their chemical classes in both the study groups, we found that the aldehydes were the dominant class, followed by hydrocarbons, alcohols, ketones, aromatics, terpenes, heterocyclics, and then phenols. Only exception was esters, which was represented by 9 species in samples from patients, whereas only by 3 in controls. The upregulation of esters in samples from patients could be because of the biochemical changes caused by the cancer causation. Similarly, while comparing the peak abundance of these compounds with occurrence above 30%, 9 of them appeared at significantly higher levels in the patient group than in the controls. These compounds were 2 of the ketones, namely 2-pentanone, 2-heptanone, 4 of the aldehydes, 2-methylpropanol, 3-methylbutanol, benzene acetaldehyde, and 4-ethyl benzaldehyde. Similarly, 2 alcohols, 1-propanol, and 2-ethyl 1-hexanol, and phenol. Whereas 2-butanone and p-xylene showed lower abundance in the patients than in the controls. Therefore, these compounds would be the potential biomarkers of gastric cancer. In conclusions, we can say that the preliminary results obtained in this study shows that gastric juice is promising fluid, providing information on potential biomarkers of gastric cancer. More samples are needed to confirm these results. The compounds identified from the gastric juice can assist in developing non-invasive tests for the detection and diagnosis of gastric cancer. And to the best of our knowledge, this is the first study reporting the composition of gastric juice volatilome in different human populations. This work has received funding from the European Union's Horizon 2020 VOCs project and a postdoc Latvia project co-funded by European Regional Development Fund. Thank you so much for your attention.
Hi, I'm Brett O'Brien and I'm a lead analytical scientist at Alstoe Medical. The World Health Organization estimates that 3.2 million deaths were attributable to indoor air pollution in 2020, with inflammation being central to the pathophysiology. COPD, heart failure, stroke and lung cancer are just some of the associated conditions. At Alstoe Medical, we're on a mission to develop a breath test for early detection of inflammatory disease using breath biopsy technology. To achieve this, we aim to identify volatile organic compound, or VOC, markers of inflammation on breath. One of our related studies is Candle Study 1, or CS1, which involved collecting breath from asthmatic patients who had been exposed to emissions from inflammation-inducing candles. Using thermal desorption, gas chromatography, high-resolution mass spectrometry, or TDG-CMS, in electron ionization mode, and Compound Discoverer software, 15 unknown molecular features were found to be markedly altered due to candle emissions. The primary focus of this poster is to highlight the utility of analyzing breath in chemical ionization, or CI, mode to aid structural identification and elucidation of VOCs on breath. Electron ionization mode often does not produce a molecular ion signal, whereas CI often does. This is because CI induces less fragmentation of the parent molecule, as shown in Figure 1 of the poster. The molecular ion reveals the molecular formula of the unknown VOC, thereby increasing the chance of successful identification. In this work, we explored the impact of reagent gas flow rate, electron energy, and ion source temperature on the production of molecular ion as well as MS sensitivity. We found that molecular ion generation is enhanced by using higher reagent gas flows, higher electron energy, and lower MS source temperature, as shown in figures 2, 3, and 4, respectively, on the poster. Importantly, higher reagent gas flows reduced the signal-to-noise ratio of the total ion chromatogram, and lower MS source temperatures are known to increase contamination buildup. These factors should be considered in addition to molecular ion generation when optimizing a CI method. Although not the central focus of this work, retention indexing was found to be of great value for peak alignment between samples and standards, particularly for closely eluting stereoisomers. Our CI approach permitted detection of molecular ion for 8 of the 15 VOCs of interest on CS1 breath, which substantially expedited their identification. An example chromatogram of one of these molecular features on breath is shown in Figure 5 of the poster. The VOCs identified in this exposure work are currently being evaluated for their potential utility as inflammatory markers in the clinic using a new targeted breathomics approach. Thank you very much for your attention, and if you have any comments or questions, please contact us at breathbiopsy@alstone.co.uk.
Biopsy conference audience. This presentation is about detection of hexanal as a volatile organic compound biomarker of cancers using the nanocomposite of gold nanoparticles and selective polymers. I am Marziyeh Mousazadeh from Tariq Modares University. Volatile organic compounds are existed in different biological samples like breath. They are used for detection of different diseases like cancers, metabolic disorders, and microbial or viral infections. They are mostly detected by gas chromatography. Here I'm going to introduce a novel and fast method for VOC detection. In this method, molecularly imprinted polymers and gold nanoparticles will be mixed and drop-casted on the surface of interdigitated electrodes. In the presence of hexanal as the template and the analyte for sensing, the MIP will swell and the current of the system will change. As you can see, the liquid hexanal will be inserted into a steel chamber, and after evaporation and becoming homogeneous by a fan, it will be transferred to a second chamber which has the modified interdigitated electrodes. In the presence of different concentration of hexanal, the changes in the current will be monitored and recorded by a potentiostat. Here, the linearity curve of the sensor is presented. It has a good linearity from 2.5 to 300 ppm, and the calculated LOD was recorded to be 1.1 ppm. This sensor was very fast. The response time was only 3 seconds and the recovery time was 39 seconds. In the selectivity test, hexanal was detected 2 or 3 times more than other VOCs, and also by an LDAPCA analysis, the hexanal was completely distinguished from other VOCs. For testing this sensor in a real sample, liquid hexanal was inserted into different biological samples like cell culture medium, serum, plasma, urine, and saliva. After 24 hours of incubation, the gaseous hexanal in the headspace of these biological samples was recorded by the sensor, and as you can see in this picture, this system was able to detect hexanal in the real sample. So VOC detection sensor based on the molecularly imprinted polymers and gold nanoparticles composite is fast, sensitive, and selective. Thanks for your attention and feel free to contact us if you have any question.
Hello, I am Robert Rintoul, a clinical academic at the University of Cambridge and Royal Papworth Hospital, and I'm the chief investigator for the evolution study. I'm working with the Alstone Medical preclinical and clinical teams on early detection approaches in lung cancer. Lung cancer, the leading cause of cancer death worldwide, has poor outcomes. Currently, the 5-year survival rate for all comers is around 16% in the UK. The main issue being that around 3/4 of patients present with advanced stage disease. However, if lung cancer can be diagnosed at an early stage, treatment with curative intent can be offered. Outcomes for early stage disease are much better, ranging from 70 to 90% 5-year survival. At present, the main tool for early diagnosis of lung cancer is CT scanning. However, access to CT for early diagnosis is currently limited, although recently the National Screening Committee have recommended to the government that national programmes for lung cancer screening should be established. The development of a breath biomarker for the diagnosis of lung cancer would be an attractive, non-invasive approach that may be more affordable and accessible. Because breath is a complex sampling matrix, high background levels of volatile organic compounds and inconsistent sampling methods have limited progress in the development of a clinically relevant breath test for lung cancer. One potential solution is use of EVOC probes, the administration of a molecular probe responsive to a disease-specific metabolic pathway, resulting in a product which can be detected on the breath. Here, we explore the use of D5-ethyl glucuronide, a substrate for beta-glucuronidase, an enzyme found in the lysosomes of healthy tissue cells. Because D5-ethyl glucuronide has low cellular permeability, it cannot penetrate into the cell to be metabolized by beta-glucuronidase in healthy tissue. However, in solid tumors, it has been shown that beta-glucuronidase is expressed in the extracellular space and therefore is accessible to the probe, resulting in the release of D5-ethanol, which can be detected on breath. And these pathways are shown in figure 1. In figure 2, we've confirmed the extracellular activity of beta-glucuronidase in lung cancer cell lines compared to normal fibroblasts, and in patient-derived xenograft tumor models compared to normal mice lung tissue. In 2019, Lang et al. Published the use of D5-ethyl glucuronide as an EVOC probe in mice, showing a clear difference in D5-ethanol formation between tumor-bearing mice and healthy mice. On the graph in figure 3, you can see correlation of D5 ethanol labels with increasing tumor size. In contrast, no or very low levels of D5 ethanol were observed in healthy mice. To assess if this mechanism is also present in human lung cancer, immunohistochemistry was performed on multiple lung cancer samples and normal lung tissue. We observed the expression of beta-glucuronidase in the tumor microenvironment of all types of lung cancer and at every tumor stage, including stage 1 and 2, which is encouraging for an early detection approach. We did not observe any extracellular expression in healthy lung tissue. Recently, we've designed and initiated the Evolution clinical study, the outline for which is shown in figure 5. Phase 1A of this study is a safety assessment in healthy controls, and Phase 1B, a dose optimization study in subjects with and without lung cancer. Phase 1A, completed in 2021, had a very favorable safety profile without any concerning adverse events. In Phase 1B, we're determining the optimal dose and time point for breath sampling after probe administration. Interim analyses will be done during this phase to determine if dose has to be changed or if a different time point for breath sampling selected. So far, we've analysed the breath samples of 6 healthy controls and 8 lung cancer patients, all with early stage cancers. Figure 6 shows the use of 2 batches of the probe. In the original batch, we examined 1 lung cancer case and 6 healthy controls. Very low or no signal was detected in the healthy individuals, with a higher response in the lung cancer patient. Some background signal in healthy subjects was the result of an impurity in the probe. A new purer version of the probe has now been synthesized and administered to 6 more lung cancer patients to date. In conclusion, through a combination of in vitro, in vivo, and clinical work, we've produced initial evidence to support the application of D5-ethyl glucuronide as an EVOC probe to evaluate non-invasive breath testing as a means for early detection of lung cancer. We've shown that D5-ethyl glucuronide leads to production of D5-ethanol in the presence of lung cancer. We've evaluated the relevance of the molecular pathway targeted by D5-ethyl glucuronide in samples of lung cancer and have early data showing that D5-ethyl glucuronide is safe and acceptable for use in patients.
Hello everyone, I'm Kenta Iitani from Tokyo Medical and Dental University, Japan. It is a pleasure to introduce our poster at the Breast Biopsy Conference 2022. The title is Screening Diabetes Mellitus through Monitoring Lipid Metabolism by Measuring and Imaging Breast Acetone using bio-fluorometric gas sensors. The human body produces energy by metabolizing fatty acids when blood glucose and glycogen are not available. Ketone bodies are produced in this process and exist in the blood. Acetone, one of the ketone bodies, has high volatility and is exchanged from blood to the exhaled breath in the lung at a ratio of 330 to 1. Particularly, patients with diabetes mellitus who cannot utilize glucose as an energy source emit more than 1 ppm of acetone, which is higher than healthy people at rest. To our best knowledge, highly sensitive and selective acetone gas sensors having real-time measurement capability and non-reactivity to humidity in biological gases are not available on Earth. So, we have been developing gas-phase biosensor systems with the help of high substrate specificity of enzymes. The secondary alcohol dehydrogenase called SADH is one of the NADH-dependent enzymes. When SADH catalyzes the reaction of acetone, coenzyme NADH has fluorescence, is oxidized, and the fluorescence disappears. By using this enzymatic reaction, we developed a fiber optic acetone gas sensor and acetone gas imaging system. The fiber optic acetone gas sensor was constructed with an ultraviolet light-emitting diode, optical fibers, a flow cell, an SIDH membrane, bandpass filters, and a photomultiplier tube. The SIDH membrane was an air-liquid diaphragm of the flow cell that allows continuous measurement of a gaseous sample. The middle right graph shows characteristics of the acetone biosensor. In fact, the change in fluorescence intensity was negative, but we showed the amount of fluorescence change instead of raw data in order to enhance visibility. The amount of fluorescence change was according to the concentration of acetone. Our sensor enabled us to quantify 20 ppb to 5.3 ppm of acetone, which range was including the breast concentration of healthy and DM patients. By using developed sensor, first, we measure the dynamic change in breast acetone concentration caused by an exercise in fasting state. As shown in the graph, the concentration of breast acetone increased by reacting exercise activity. We would like to mention that all human subject experiments are approved by the IRB of Tokyo Medical and Data University. After that, we investigate the possibility of classifying DM type 1, DM type 2, and healthy subject by measuring breast acetone. As a result, we were able to distinguish not only healthy and DM patients, but also DM type 1 and DM type 2 with significant difference. We would like to emphasize DM patients are already getting treated at the hospital. Therefore, blood glucose levels were not so high. We believe that if we had someone with hidden diabetes, be able to find them by using this sensor. Another topic of our poster is gas imaging system. The imaging system was constructed by expanding the bio-fluorometric principle into two-dimensional. Acetone gas imaging was possible by capturing the spatial temporal change of fluorescence on the SIDH membrane as shown in the images. The experimental data indicated that image-based quantitative gas sensing was possible as well as fiber optic gas sensors. We achieved monitoring a gradual change in glucose metabolism to lipid metabolism by the system. By the way, acetone is contained not only in breast but also emanated from human skin. We have continued to develop the gas imaging system to achieve imaging of gaseous acetone emitted through the skin. Let me summarize our poster. We developed highly sensitive and high selective acetone biosensor. Also, spatial temporal imaging of gaseous acetone was possible by using bio-fluorometric gas imaging system. By using that system, real-time monitoring lipid metabolism and DM classification was demonstrated. Thank you very much for your attention. If you have questions or comments, please post them on the page. Also, do not forget to vote if you're interested.
Thank you for listening. My name is Wout Duthoo. I am working as an R&D project leader for IMEC, a Belgian R&D and innovation hub for nanotechnology. In this poster presentation, I would like to present to you the work that IMEC has done in developing a fast point-of-care PCR test for infectious diseases, which is based on a novel way to capture aerosol from exhaled breath and run molecular diagnostics on this breath sample. Today, we have proven the value of this technology for COVID-19, but we are now actively looking to expand our menu to other respiratory infectious diseases that can be detected in breath. Like many other innovations, it's actually the COVID-19 pandemic that sparked us to start working on this, as it confronted us with the limitations of existing diagnostic technologies. To overcome these limitations, we come up with a new concept for a breath test. This breath test needed to be reliable. It also needed to be more comfortable than the traditional nasopharyngeal swab, and also turnaround time had to be drastically reduced. Since we would capture and detect the virus in exhaled breath, which is exactly there where it is known to spread, we also hoped that this test would allow us to detect infectiousness rather than infection. In order to get there, we of course had to develop the technology. In this case, the technology is a new micro-machine silicon sieve that would allow us to capture the small aerosols in the breath before performing a qPCR on that chip. The chip itself, zooming in on it, the principle will become clear. A chip works as a miniaturized sieve, a sort of maze for air, so to speak. The holes in this sieve are slightly misaligned so that the air needs to make a sharp turn to pass through the other side. Smaller particles manage to make that turn and go through, but the bigger particles from 300 micron onwards collide on the back of the sieve through impaction. These bigger particles may contain viral RNA, which we assumed could be detected using a molecular PCR technique. And in next clinical study, this is exactly what we set out to prove. We collected samples from 32 positive patients in the hospital, as well as 22 healthy volunteers. All healthy volunteers tested negative both on MP-PCR as on breath, learning us that the negative percentage agreement or specificity was 100%. Positive patients confirmed positive by means of the golden standard MP swab. Out of those 32 positive patients, 24 also tested positive on the breath, whereas 8 tested negative on breath. This amounts to a total positive percentage agreement or a sensitivity of 75%. However, we've learned that this is actually close to 100% in the first days of infection. Indeed, in a follow-up second clinical study, we screened high-risk contacts of known infected individuals. The aim here was to have insights into day 0, which is the first day a test would indicate that a person was infected. We then followed up those patients up to 7 or even 10 days following that first test. Zooming in on the results, you can clearly see that the sensitivity of the qPCR on breath superseded that of the rapid antigen test performance and was equally good or even better than the performance of the MP swab in the first 2 days of infection. From day 3 to day 6 onwards, all tests performed equally well, but importantly, from day 7, you can see that the breath test quickly turned negative, way before the other tests, which would then stay positive for days or even weeks to come. In a recent paper, we have claimed this pattern is compatible with an infectiousness test. Importantly, all the studies I've mentioned above were done with a proof of concept version of the technology. In the so-called non-integrated workflow, a lot of manual steps still needed to be done in order to obtain the PCR results. Remember that we actually wanted to make a fast point of care test, which is why we also spent considerable time and effort into actually designing, developing, and building an integrated workflow. This integrated workflow consists of a breath sampling device, which would allow us to collect the sample, then fill and seal the silicon sieve in the breath sampler itself, and insert the device into a qPCR machine, allowing us to have a sample to result time close to 15 minutes. In a last clinical study, we actually validated that this integrated workflow works equally well as the non-integrated workflow we used to obtain all the results we've discussed in the clinical studies above. To sum up, we have developed a new technology to enable aerosol collection at the point of care in a fast, comfortable 1-minute breath sample, and then combine the sampling method with a sensitive molecular backend. We have validated this technology for COVID-19, and we are convinced that it will have value for other respiratory infectious diseases, bringing exhaled breath diagnostics closer to the clinical practice. I thank you all for listening. In case of questions, do reach out to me on the email address shown on the slide. Thanks.